Okay. So we will start with we will start once again with another tutorial, again in isobaric labeling, but now we switch to a different kind of tutorial. So just briefly what we did before, we saw a tutorial of how to set up your correction factor in isobaric labeling with MaxQuant, and then how to analyze this data in Perseus. I introduced that now it's possible also to run TMT frames in MaxQuant. Again, I'm not showing you here how to do that, but we will soon publish a paper regarding this, so you will see there how to run and how looks like, let's say, the results of using TMT frames in MaxQuant. But what I will do with this second tutorial is to show to you how to use the PSM weighted normalization that Christoph during the isobaric labeling theory introduced to you. Just briefly, the theory behind. So we define this PSM weighted median normalization to remove the batch effect that we have our TMT datasets. Because the problem of the MSN, MS2, MS3 is exactly one of the disadvantages is exactly this batch effect. And for that reason, to date, let's say in the last few years, in many labs, the idea was to define, in addition to the sample channel that you have in your TNT set, to define a reference channel. A reference channel that is a mix of the samples that you have in across in your TMT set, and this one is exactly a sort of a control that you can use it for the control the technical variability that you have in your analysis, and you will use it exactly in your normalization, like in case of MaxQuant, the PSM weighted normalization. This one should reduce your normalization. In our case, the PSM weighted normalization in MaxQuant reduce the batch effect. So as exactly in the Umap that Christoph showed you in the theory, you should move from a Umap that is clustering based on the batch, from the batch to the biological sample that you have. And again, this is the paper that we refer, also Christoph showed to you before, and there it's explained exactly also the mathematical theory behind this normalization. As I did it before, I will introduce you the goal of this tutorial. So what are we going to do today? Again, we will take one data set, we will see a paper, we will see the data set defined, we will see the experimental design, but what are we going to do? As similar like before, we will do that first in MaxQuant and then in Perseus. And we run once again a TMT dataset. In this case, we don't have any correction factor, so we don't stay focused on the correction factor, but we will see exactly how to set up this reference channel in case you have this kind of data set. But most important, I will also show you not only how to use it as standard one, so you set the reference channel as it is in the experimental design that is in the data set, so how they show to you in the metadata. But also I will show to you using the PSM weighted normalization that is in MaxQuant in different ways, because you will see that the results then in Perseus will be different. And for that reason, you see this option, you see that we will use the PSM weighted normalization using the reference channel as it is in the experimental design. It means that, let's say that you have TMT templex and the last channel is the reference channel. You will tell to MaxQuant that that one is the reference channel. And this is the option 1 here that I listed. The second option 2 is to do the opposite. So taking the same example, we have a TMT templex and the last channel is the reference channel. We will not tell to MaxQuant that one is a reference channel, but we do the opposite. So we will tell to MaxQuant that the others, the sample channel where we have tissue, cell lines, whatever the data set is, are our reference channel. The third option is without using a PSM normalization. So, okay, we have a reference channel, whatever, we don't use it. What we should expect is that we still see the batch effect because we didn't apply any normalization from this one. So it means that we have this free option, and it will be 3 different MaxQuant process. So it means that with the same data, you will process the option 1, setting the reference channel as it is in the experimental design, and you will process, you will get an output, the one TXT folder. So let's say TXT folder from the option 1. Then you will reprocess, taking the sample channels, and you will get another output from a different MaxQuant process, and then you will do that without the PSM normalization, so that you can see from 3 different MaxQuant process, you will see the difference across this, and then you can see exactly how it works, the normalization, and how effective it is. Same, from these 3 different output, you will get 3 different MaxQuant output, 3 different TXT folder. So it means that if we think about the protein groups TXT output file, you will get 3 of them. What I will show you is that we will take one of them, and I will show you how to do the analysis of that one. And if you reproduce exactly the same analysis with the other protein groups, using exactly the same parameters and everything, you will see exactly the difference without using and setting a different reference channel in MaxQuant. Before to show you exactly how to run this in MaxQuant and then do the analysis in Perseus, let's go through the data that we use. As I did before, we started to prefer to exactly take a paper, see which data that you use, the experimental design, so that you have a real example of this, and then we can see how we use. And here in this case, we took this data set. This is TMT MS3 templates. And as you can see, yes, there are different tissues. So there are human body, proteome map. And here, for example, in the figure B, you can see one TMT set where you can see that there is 2 reference channels. And then there are different samples. So the first channel is the reference channel, the last channel is reference channel, then the samples. If you read this paper, and if you go to the supplementary material, they provided to you the metadata of this. And here is a part of it, just to give you an idea. So 1, 2, it's exactly 1 and the 2, so the red one, the green one, sorry, and then the orange one are exactly this TMT set. So the first one and the second TMT set, you can see the channels, and then you can see exactly which are the references. The one that here I colored in yellow. But you can see that then you see that there are across different tissue, as some of these samples are also repeating different TMT set. Like you see muscle, we have in the second channel in the first TMT set, but then you have twice in the second TMT set. And again, so you see just only these 2, but there are 56 of them. Yes, you can see. So there are, yeah, here they call it run, because in the paper they call it run, but you have basically 6 TMT set. So we have this experimental design, and we have to do exactly the 3 options that I said to you before. So here graphically, what are we going to do? We don't apply normalization, but we have this reference channel. Okay, we can set the reference channel, but what MaxQuant will do is that if you don't set up the weighted normalization, you can say which is the reference channel, but anyway, MaxQuant will not apply it. So it's the first option when the normalization is not applied. The second option, the one that I colored in orange, is the one that is with the reference channel or reference channels, because like in this case, there could be more than one. And MaxQuant will take this reference channel, and then we'll do the ratio across the other sample to normalize them. The other one is the one that I started to explain you before. So instead to tell to MaxQuant that we will take the reference channel as it stated in the experimental design, we do the opposite. So we take the others. What MaxQuant will do is that in the first option, as I said, it will not do any normalization because we didn't set up any normalization. In the second option, the orange one, what it will do is that it's exactly normalized based on the reference channel as it is. But then in the third one, there is, because there are more reference channels that you set up, what you will do is sort of, you will create a reference channel, a sort of in silico reference channel based off the median across all the samples that you set up, that there are there, the one that you selected as reference channel. And then that one, that here is colored based on the samples that are there, that one will be used as reference, and then it will apply the ratio across all of them. And yes, and as I said before, so what it will happen is that we will do this one is 3 different process. So it will be one process in MaxQuant, another process without the normalization, another process using the normalization reference channel, then the sample channel. And as there are 3 MaxQuant processes, then there will be 3 different downstream analysis in Perseus. All of these, these 3 process is, as I said, is in the 3 different MaxQuant process. But in the USB drive, you find, because there are 3 different MaxQuant process, 3 different MQ path file. So you will see when you use the first one, when you use the second one, and when you use the third one. But for Perseus, what we did is that we imported everything in one Perseus session. So you will see these 3 different output, the protein groups file in the same session. So you will just see 3 uploads. And then you will see that for each of 3 uploads, you see the workflow that it was done exactly the same for all. And then you will see that the results is completely different. Let's go to MaxQuant. So here I'm showing you of how we will start with the option as it should be, because we in the experimental design, they say that we have a reference channel. So we start with the option orange. So as is stated, in the experimental design, we know that there is a reference channel. So let's do that as the experimental design says. So in MaxQuant, what we will do is that we import our raw data, we set the experiments, and yes, here, yeah, you don't see correctly, it's only you see label instrument, but I labeled exactly as it is labeled in the experimental design that is in the paper, because again, as I said before, if there is a metadata, please follow the same label, the same labels that they give in the there, because it's always better to reproduce it and then go back to your analysis. Here, sorry, you don't see it because it was extremely long, the labels, so I needed to reduce for the screenshot. But as I said, we do follow this as the experimental design. I showed you before that in this case, we have 2 reference channels. It was the first one and the last one. It's a TMT-plex. So what we say is that we load our raw data, we set the experiments, and we set the reference channel, and we have the templex. So the first one, the last one means that we select 1 and then 10. Very simple. Here you see, again, you see only 4 raw data. If you go to the paper, you will see that there are more raw data because it's a bigger one, but I'm sorry, we could not provide all the raw data in the USB drive. You can download whenever you want. However, the process, the protein groups file, so the output that is in the USB drive, it's the one when we process all the raw data. So the raw data provided the pure raw data are just only 4, the one that you see here, but the up to from MaxQuant, all the raw data that are in the private repository with this paper, all of them they were used because we needed to reproduce exactly this human body, this proteome map that they explain in the paper. Okay, we set our reference channel. Let's go to the quantification. So this is similar as we did it before. So before we were talking about a TMT MS2 Pro, TMT Pro MS2 16-plex. Now we have MS3 10-plex. So what we do is that we do reporter ion MS3 and we select 10-plex. We don't have any correction factor, so we don't do any editing in each channel because we don't need to do that basically. But which is the difference now is that we have to say that we want to apply weighted normalization. And here down below, you see normalization, we need to select weighted ratio to reference channel. By default, MaxQuant, in this option, it's none. So it means that you don't do that. So be careful to do that, because maybe you say here the reference channel, but here you don't select weighted ratio to reference channel. The weight normalization will be not applied. Okay, so if you do this one, but you don't change this parameter normalization, the weight normalization will be not applied. So this is extremely important. And yes, so pay attention to that. Please do that. You need to add, as usual, FASTA file. Here you saw from the previous slides that is just human body. We don't have any mixed shampoo, mixed species, so we don't have to define any taxonomy or ID and so on. We just need to include the FASTA file. The FASTA file that we just downloaded from Uniprot, Homo sapiens in this case. And we just start, as you saw many times, as I showed you before, as they show you last Tuesday, how to process, and always, once again, remember to change the number of threads based on the memory that you have in your server where you run your dataset. Okay, so this one is how to process when we follow the experimental design as it is in this case, where we have the first channel and the last channel as reference channel, and we want to use them. We want to use them for a normalization, and we use the PSM weight normalization that is in MaxQuant. So we do that as it is stated reading the paper. But so we do this one. So the reference channel as it is stated, the orange one. Let's say that I want to do the other one, the one that I was saying that is the opposite, okay? So it means that if I go back to the metadata, is that in this case, I will exclude completely the reference channel. So the first, or better, I will always say that is templex. But what I have to say to MaxQuant is that take the sample channels, that in this case is from the second one to the ninth one. So the one that I highlighted with this rectangle in red. So the difference will be compared to the MaxQuant process that we saw before, is that we always do say to MaxQuant to do the weight normalization. So always do this one, always the weight normalization, reporter MS3, templex as usual. But the only difference will be this one, is that we will say to MaxQuant the sample channel to take that one as reference channels. And this one again is another process. So this one you will do that you will open once again MaxQuant, you load your data as usual. Just this one is a difference, you process. That's it. The third option was the one where the normalization is not applied. How you do this one? Just you take it as default, and the default, as I said before, is none. So when this one you keep it as default, that is none, the weighted normalization is applied. You can put whatever you want in the reference channel, you can put all of them, the weighted normalization will be not applied. So this is how to do that in MaxQuant. So we have these three different processes, these three different MaxQuant. Now we have to go to Perseus. And since I started with the one that where we follow how is the experimental design in the paper, so as the reference channel as stated, I will go through that one. So I will use the example of how it is done in the paper. So if we go in Perseus, what we need to do is that we need first to take all our channels. We don't care in this case the corrected, so you will see that there is your output also the reporter intensity corrected, but we didn't apply any correction, so there is no difference between the reporter intensity corrected and the reporter intensity. So just take the reporter intensity. As you can see here, there are many of them, because if you look at the experimental design of this paper, you saw also before that there are 56 TMT specs, so for each TMT set, you have 10 reporter intensity, so it's very big. But you need to import all of them, because we use all the TMT set to reproduce this human body. Okay, so then you have to do, you need to do basic filtering. This is something that Juan spent time this morning to do that. So remove the reverse, the all identified by sites and the potential contaminants. So this is the usual filtering that we do also in this case. That Juan this morning did it step by step, opening Perseus. So this is exactly the same, nothing changed. What we do is that we log transform our data. So all the report intensity just using base, this is the transform is under basic transform, and also Juan show you this this morning how to log. We can do log 2. So this transform can be any kind of transform. In our case, we need log 2, but here you can change as log 10. You can do also an exponential, but in this case, we need log 2. But what we need to do also is that we need to say to Perseus, again, we need to include an annotation. We need to include an annotation because we need to say MaxQuant, sorry, to Perseus, the reference channel, the sample channel. So again, we have to take this metadata that they should provide together with your paper, with the paper, of which, what is that sample channel, so which tissue, which cell line you have in that channel, if that one is a reference channel, if that channel is empty. So all kind of information that you need. Again, here we have many reported intensities. So we have 56 TMT set multiplied per 10, so it's very big. So this is the number of the reported intensity that you have. You can do that one by one, but you will spend, I don't know, at least 30 minutes just to do that one by one manually in Perseus. So again, what we can do is that write a template. And you can do that under annotation, the categorical annotation row, write a template, and it will generate again a TXT file that you can remit as Superfair. By default, here is groups. What you can do then, go to Excel and import all the information. This one, I just copy and paste the Excel file that is together with this paper. So if you look at the paper and you go to the supplementary material in the paper, you will find as first or second TXT file exactly this one. So all the information that you have is just copy and paste. Also because I labeled all the sample as it is in the metadata, it was very easy for me then to recognize each sample to which one is referred. So that's why, again, I highly suggest you to, even if it's a very long label, sometimes maybe you don't, it has some definition that maybe for you it's not clear what is that, but that one at least with the metadata, then you can match each channel that you have. Again, you do this one, and when you've done, save it, actually also close it, because if you keep it open, then Perseus will ask you to close it. So please save your TXT file, close in the Excel, and then you read the file once again. You give the location where you locate it, and then you import it, and you will see all this information as annotation. As you can see from the experimental design, also here, there are the reference channels, and sometimes here in this screenshot it doesn't see, but there are also some channels that are called blank, that are basically the empty channel, because in this case, some channels, they kept it as empty channels. But we need to remove them, because the reference channels, or also the empty channel, we don't need it, because the reference channel, yes, we need the reference channel, but we need the reference channel when we use it MaxQuant. MaxQuant we already processing MaxQuant. Now what we need is only our sample to see exactly if we can see if this normalization worked. So we need to filter, we need to remove the columns that are our reported intensity. We need to remove the one that are referenced and the one that are empty channels. How we do that? We just filter based on columns, and taking the annotation row name, the annotation rows that is called the tissue name. And it's called tissue name because in the experimental design, in the metadata, it was exactly called tissue name. We select the reference that are the channel that are the reference, and the blank that are exactly the empty. In this case, compared to how we did it before I was telling you always keep the matching rows because we wanted to keep only that one. Here is the contrary. We wanted to remove this one because we don't need it. So we keep the mode as remove matching columns. So what Perseus will do is that we will remove this one and we will keep all the others that are the samples, all the tissue that we have. Okay, and it will reduce the matrix. Then we need to filter our data based on percentage. So we always receive opinion, which percentage should I use? Should I use 70, 75, 100%? We always say that it depends on the experimental design, and that is true. So first, if they say in the paper, do that as they do that in the paper, because yes, at least in the additional methods or in the supplementary material, they should also tell you how they did it. And so there it's written. Or what you can do, if it is your data, if it is something that you don't reproduce something from a paper, but it's just your data, maybe what you can do is exactly play around that. So like here it's 70%, then maybe you can go to 75 or do 100%. So you remove all possible protein groups that they have at least one missing values, and you check if there is a difference. Actually, in this case, I provide 70% like here, but also providing the Perseus also 100%, and you can see that there are not so many differences. So that's, but I did it exactly to show you that you can play around that, especially if it is your data. If there is something from the paper, you reproduce it as it is. If it is your data, first you play around, and if you see some difference, you need to check which are the protein groups that are removed and why these protein groups makes so much difference. So you will look at what are these protein groups. You go to UniProt, see the UniProt and why this one is give difference on in your data overall, and maybe yes in specific biological sample as well. Here in this question is 70%, so we apply this percentage as 70%. Sorry, this one. So now we have this big data, and we need to see the difference across these different runs in MaxQuant. I showed something similar yesterday with MaxQuant Atlas, that there was this high dimensional data. We needed to reduce it in a low dimensional data. Okay, and what we do that in order to reproduce it is that you can apply a dimensionality reduction. But what is the problem with when you apply a dimensionality reduction is that you needed to remove any kind of missing values. So which are the options? The option could be that here you filter based on 100%. So it means that if there is at least one missing values, or that protein group will be removed. Or if you do like here 70%, there will be some missing values, but you need to impute this data, because we needed to have, we did not to have any missing values. What we do in this case, so MaxQuant Atlas, we did it a specific imputation, but that one is because we have an atlas with different data set, different dynamic range. Here we have just a basic imputation based off normal distribution. This is something that also Juan showed you today when he was showing you the PCA, and it's just basically the same one. So just apply an imputation. We need to do that in the Umap analysis. So we need, or you can also do that in PCA, but we will do that again in the Umap analysis. So what you need to do is that go to under clustering a Umap analysis and use Umap analysis. But as you can see from this screenshot, there is this section that you see generic executable, R script and something. Why? So Perseus is in C#, as it is in MaxQuant. But thanks to Jan Radolf, PhD student, he was at Jürgen Koch's lab, now you can integrate your R script into Perseus. So you can, first you can do your plugin. So you have your standard Perseus, and you can develop the plugin that then you can integrate in Perseus, because you have some functionality in Perseus, but we don't have everything, okay? because we define the standard one, but maybe you need something specific, someone else needs something else, and we need also as well something specific. For that reason, the idea is that we provide you the standard to do just basic downstream analysis, but at the same time you can develop your own Perseus plugin. How you can do that? You can do that in C#, and there are many examples in our GitHub repository. If you go in the Cox Lab repository, the Perseus plugin, there are some of them already done in just pure C#, and you can do that like that. But another example is that, like, look, I don't want to develop from scratch entirely in C#. I want to use just an R package that is in R or in Python. Okay, I just want to call that R package. So you will install in R that R package, and then you will call it with the library parentheses the name of the R package. You just want to call a function, and then but you want to use it, and you want to use it not in RStudio, but I want to use it in Perseus. So it means that I have my matrix. I want to use this matrix in a way I want to take this matrix, run it with this function of this specific R package, and then when this one is run, give me the output automatically in Perseus. This is something possible. That we have a paper about that that is explaining you exactly step by step of how to do this. But this is exactly an example. So this Umap analysis, we did exactly this. So it means that you have your matrix, that it was long transform, it was filter 70%. Now what we do with this one is that you have from matrix, you have your matrix, we take it, we call our package Umap in R. We apply this, we apply the function Umap that is in this R package. We calculate R, and we get the output, and this output will be automatically a new matrix in Perseus. This is available. Also, it's available in the GitHub repository. So you will see this one Umap analysis, so how we developed exactly that, and it's also with others. So there are many examples to reproduce that one. So it means that we did it for Umap. We did also actually for Disney, but you can do that for many others for the R package that you want to use. And but there is only one thing, is that if you want to do that, you can develop as you want, but you need to install Perseus R. And Perseus R, it's an R package that Jan Radolf developed in a way exactly to read this R package that you want to integrate in your analysis. So it means that you go to Visual Studio, you developed your plugin, fine, but then when you generate it, if you want then using Perseus, you have just to go to R, to R script, yes, to executable R script, and install Perseus R. As you install our other package in RStudio, you need to install this one. I'm not showing you here how you can do this one, but there are many YouTube videos where we provide this one. And actually, also in the PDF at the end, we also included exactly which step you need to do that to install this Perseus R before to use any Perseus plugin where R or Python is needed, like in this case. So it means that, like in this case, that you have Umap analysis and you see that it needs the R script. So when you see such kind of activities in Perseus, is that okay? First, before to use it, I have to go back to R and install Perseus R. How to do that? There is a specific YouTube video for that. And we also apply, and you have also the PDF, just few commands, just copy and paste that one, and then it will go. It is very straightforward. If you don't do that one, if you don't install Perseus R, this bottom that you see in green, sorry, will be red. Because this executable is there exactly because in a way that Perseus check if you first install Perseus R, because if you install it, I can do this Umap analysis. Otherwise, I could not do that. If this bottom in green is red, you can press OK. The Umap analysis will not work because it needs this package. So let's go back to our experimental design. We do this one, we do the Umap analysis. So what we get an output are the components of our Umap analysis. We did it 3 MaxQuant process. So we did it, we have 3 different MaxQuant output and 3 different workflow in Perseus. So what we will get is that 3 different Umap analysis in these 3 cases. Let's see how they look like. If we see here, you can see that the no MaxQuant norm is when we didn't do any normalization. When we apply the normalization based on references, when we took the references stated, and then you see the MaxQuant normalization as samples. In the first row, we color the dots based on the set of experiments. So these many batches that we have in our experimental design. But then we were like, look at the MaxQuant norm using the sample, so the one that is highlighted in purple, look at that it started to clustering differently, okay? So we were like, okay, this one maybe is clustering based on something else. We colored based on tissue, and indeed, you can see that it starts to color based on that one. And if I zoom in, you can see exactly that it starts to cluster into that one. Actually, you can also see that in this one, you can see the pointer, yes, that this one, for example, it's you see this triangle that are 2 different region of heart, yes. You see that all the biological sample are here, and actually it's also together with the muscle. And if you know a little bit biology, it's very good that they are clustering together. And at the same time, you can see also this triangle here small that are the 2 parts of brain. So we can exactly say that this one, it was the best one compared to all the others. Okay, but what does it mean? If we go back to the old case that we have, it means that the last one, when we took the sample channel, it was the best one. So I could say, why do I need the reference channel? Indeed, in this case, it was not needed for MaxQuant. So if we go back to experimental design, we can say that in this case that it was templex. These 2 reference channels that are there, at this point I can use it for another sample. So I can use these 2 channels for an additional sample instead of to use it as mixed. Because thanks to this normalization, I can reproduce my silico reference without a reference already defined during the sample preparation. And indeed, I proved to you this one here, but if you go back also in the paper, regarding the PSM normalization, Sanquan and Peli, they already did it use without a reference channel. So they also use a data set where a reference channel was not there, and they took all the sample channels as reference channel, and also in that case, showing the Umap, they show exactly the results that it was clustering very good exactly in this case. So what we would like to address with this tutorial is that exactly how to run a TMT data set with this reference channel, so to show you how to run it using the PSM normalization. But at the same time, we also realized that in case you don't have the reference channel, you can always use this PSM normalization, and actually it's working pretty well. And also using only the sample channel as is also the best choice to use it. And yes, so please follow the tutorials, because you will see exactly these 3 cases in the Perseus session, and you will see exactly this 3 Umap in the scatter plot, where you can do where you can see exactly these 3 difference, and you will realize that the tissue is clustering, that when you apply the PSM normalization based on samples, is clustering based on tissue of origin. Thank you.